That'd be hilarious if you caught that on tape.
I know anyone who's not here will be like, what? Okay. Let's get started. Who's excited about spring break? Who's excited about St. Patrick's Day? Yes! Okay. Who understands St. Patrick's Day? I don't get it. A little bit? Okay, later we'll check. Half of my son's preschool class was in green, and I was like, that's so weird. And then my kids' preschool, my girls' preschool class was like half in green. And I was like, what is with the green? What a coincidence. And then someone's like, St. Patrick's Day. Let's see, 1, 2, 3, 3. Not that many. One green sock. Okay. Business, exam 1, pickup and recitation. Most of you have gotten yours already. Those of you who have not, get it today because I'm guessing your recitation today. Also, you can take that time to ask me any questions about it. I'll try to make sure I have the exam with me. Exam 2, week of 4-1. Extra credit. The other day we had an extra credit. I only got one answer, which probably people forgot, is my guess. The question was, how is lagging strand telomere sequence replicated without a primer? Not replicated, but synthesized without a primer, right? And we'll look at this again and you can see what I mean. But the whole thing is you need that primer, right? Because you need the 3' OH, and the end replication problem is all about how with removal of the primer you can't get full replication, and then telomerase comes in and extends the leading strand, and then you still get replication, but there was no, in the textbook, there's nothing about a primase. So my question was, well, how is that happening? And I got one person who got the point that the DNA polymerase that's involved in that extension there has a primase it's involved with. And actually people have shown that there's an RNA primer there. And if you think about it, it makes a lot of sense because your leading strand's always longer than your lagging strands anyways, and the job of the telomerase is to extend it further so that it could be extended further, but you'll still have that overhang on your leading strand. We'll talk a little more about it. Okay, so quick word on memorizing versus understanding. A few people have come to me to talk about their grades, and we go over the exams, and what we've come up with is they are not studying effectively. They're trying to memorize everything, and what happens is then you sit down, you're stressed out, you're taking an exam, and everything starts flip-flopping in your head, and you make really foolish mistakes because you never understood what you were doing. You were just stuffing information in your head, and then as it's coming out, it's wrong because there was no logic to it. So I just wanted to talk about that briefly. So here's an example, because someone was like, I don't understand what you mean. What do you mean, understand? And I was like, so I gave one example, and then I thought of another example. So I just wanted to take a minute. Those of you who understand everything, well, just bear with me. But my guess is most of you could use a little bit of a tutorial here. So an example of memorizing versus understanding. Acetylated lysine and its effect on gene expression. Memorizing is when you go acetylation, activation, and that's it. And then you get in the exam and you get confused, and then you write down acetylation, repression, silencing, and it's wrong. Now, if you understood it, you would say acetylated lysine. Lysine, let me look at the structure of the lysine amino acid. Look, it's positively charged. Let me see how that charge is affected by acetylation. Look, it gets rid of that positive charge. Well, DNA is negatively charged and binds strongly to positively charged lysine. When we get rid of that positive charge, it loosens the interaction, which then opens up the chromatin so it's available for gene expression. You'll never make the mistake if you understand that. Everyone understands positive, negative bind to each other, right? You've all used a magnet before, right? So, but taking the moment to just stop yourself from the automatic need to memorize, because you've gotten this far, probably on memorizing, because 201, 309, you can get by because it's a lot of fact they're trying to get you to understand, just retain to get that background. But at this point, you really need to be able to use the information, which you cannot do if you're memorizing. Okay, so the second example is that when we're thinking about the end replication problem, right? So what is the end replication problem? Oh, I forgot my little list. What's the end replication problem? Someone who never raises their hand. Right now. Okay. I will call on Navdeep.
So on the lagging strand, you are not having the 3' OH group.
Okay, so on the lagging strand, due to the lack of a 3' OH, there's an end replication problem. Kind of. You're right, it's on the lagging strand. The problem is there's the removal of the primer, so it can't be replicated to completion, right? So that's your end replication problem. And through multiple cycles of replication, you're going to have this progressive shrinking of your chromosomes. So this came up because my grad student was saying that a professor got confused and said leading strand, not lagging strand, in terms of what DNA strand it was involved in. And I thought to myself, this is a perfect example of how you can think about it and understand it and then know exactly where the problem is, right? So you know that it's happening on the lagging strand because that's where that primer is that needs to be removed, right? Does everyone agree? Right, so you'll never confuse that because you understand that because you have replication that's discontinuous on your lagging strand and you've got these Okazaki fragments and they've got all these primers that need to be removed, that's where you're going to be missing that 3' OH and not be able to synthesize it all the way. And that's your end replication problem. If you sit there and just memorize end replication problem, telomerase, and nothing else, then you're not going to use any information. So when you understand it, you don't make foolish mistakes because many people are coming to my office, and once we talk through something, they're like, I cannot believe I mixed that up. And you don't mix things up when you get it. And the example I gave to someone else is if you're baking a cake, you understand that you need to mix your wet and dry ingredients before you put it in the oven. Would you ever just randomly put your flour in the oven? No, because logic has told you over the years that is a foolish thing to do and you won't get a cake. So it's kind of the same. If you understand all the reasons why you're studying something and why these different processes are happening, then you won't put your flour in the oven. Okay, now I'm done lecturing you. Okay, any questions about what I'm talking about? If anyone still is like, I have no idea how to study without memorizing, please come see me because it'll make your life easier if you can figure that out. Okay, so this is what we've done so far. We've been talking about mechanisms of regulation of the cell cycle, checkpoints. We've been on S phase for the past week or so. We talked about DNA polymerase. We talked about initiation in both bacteria and eukaryotic cells. We talked about origins in bacteria versus eukaryotic cells. We talked about how origin firing is regulated in both bacteria and eukaryotic cells. We've talked about priming, synthesis, and termination. We talked about decatenation and the function of topoisomerase in decatenation. And then the end replication problem. So let's just start where we left off. The end replication is solved by telomerase. Right? So this is the enzyme that has its own RNA template and extends the leading strand of the DNA. So who thought that telomerase fills in the ends of the chromosomes? I think I thought that at one point, way back in the day, where I thought like there's that primer is removed and there's this this thing that needs to be filled in, and telomerase comes and does that. It doesn't. So just make sure you don't think that, because I figure if I was misled at one point, maybe someone else here is. So this is DNA synthesis. You're synthesizing DNA using an RNA template. And that RNA template is part of the telomerase enzyme. Okay? This is where we finished the other day, right? So you have your parental strand of DNA. So this was your leading strand, and that synthesized all the way to the end. And here's your lagging strand, which has this, it's incomplete because you had to remove this primer, and there's no 3' OH here, right? And so telomerase will bind, and it will extend the leading strand further. And then what can happen is DNA polymerase will come along, and it has a primase that it associates with. So you don't see it, you don't see a primer, but there actually is an RNA primer here. That's only a few bases long. And you get synthesis and extension of the end. You still do have, this primer is then removed by RNase H, and so you still do have this overhang here. So you never have equal ends, okay? Oh, wait a minute. No. Oh, there it is. Okay. Whew. I thought I missed something there, but we're gonna be okay. Yes.
So you said that telomerase, all it does is it adds some more bases to the leading strand, and do those bases serve as kind of a replication site for the RNA polymerase?
They are a template. They are the template because RNA polymerase is going to come in and extend. See all this? This is all not RNA polymerase, DNA polymerase. No, RNA polymerase is only involved in transcription. Okay. So they're going to extend. So they're just, what it's doing is it's increasing the length of the template. So then DNA polymerase 1, I believe, comes in, or alpha, and extends that. Make sense?
Does it just extend it straight, or does it doesn't do the fragmentation?
No, because all it's doing is it has a primase. So the question is it just this continuous synthesis, or are you having something similar to what happens at the replication fork? And no, you don't need that because it's not a replication fork now. All it's doing is kind of completing the end of one strand of DNA. And so it has a primase, which will make a primer right here. And all it does is complete from here to here. Okay? Yes?
Does the extra hair stay on the end of the leading strand, or
Yep, they say they're so this is so the question is, do the extra base pairs stay on the end of the leading strand, or does something happen to them? And what happens is, do you mean these, or do you mean what's added by telomerase? Do you mean the overhang at the end?
Isn't that what was added?
Huh?
Is that what was added? Or?
Yeah. So along. So you start here, and then you get extension for quite a ways, and then you have addition of your lagging strand nucleotides, and so all of these are there. They are added by polymerization, and they're there permanently. They're not going to go anywhere. Now, if you're asking what happens to this overhang, as you'll see in a minute, is it gets tucked in. Okay. Any other questions? Good questions today. Yes. Here, can you talk a little louder?
Since telomerase is using an RNA polymer to extend it, and it all as a primer to attach to the protein, wouldn't they have the same template, like the primer would be the same?
So the question is, telomerase uses an RNA primer, and so does DNA polymerase, but different. It's very different. Telomerase, the RNA primer is actually part of the enzyme, whereas DNA polymerase associates with a primase, which creates a primer. So they're quite different, but they serve, well, no, they serve very different functions because the primer, really primer. I guess the primer that's in the telomerase is a template. It has an RNA template. So think of it as a template rather than a primer, because a primer you're thinking of is because you're extending from there, because it's supplying that OH group to add your additional nucleotides. That is not what happens here. That's not what's happening with that, right? That's just a template. Okay? Does that explain your question or answer your question? The sequence would be the same.
The sequence of 2 primers.
Would the sequence of the 2 primers be the same? You mean this template and the primer sequence be the same? Well, in a way, yes, because you're repeats, because this is a permanent sequence in a way, right? Because it's part of the enzyme, and so it gets over and over again along the strand. And then your primer, your primase, all it does is do the complementary sequence to that. So it would be similar. It would be the complement. Yes.
So are those last bit of DNA going to be degraded since it's single-stranded?
So it's gonna fold over, and you'll see that in a minute. Yeah. So the question was, will that overhang be degraded because it's single strand? You're absolutely right. If you leave it single strand and hang in there, it will be degraded because it's not protected. And what happens is it folds over and actually binds in between the DNA duplex, forming a T loop, which I'll show you in a minute, and that stabilizes it. Okay, any more questions? Okay, so let's just talk a bit more about telomerase. Telomerase processively elongates telomeres by using its own RNA primer as a template. I think primer is not the way. I think just we'll call it a template. So here are some thoughts about telomerase. Has anyone noticed that the structure of telomerase looks similar? What does it look like? Does it look like something else we've seen before? Okay, so people are going like this, right? It looks like a hand, just like DNA polymerase. So when you think about proteins and their structures and how structures relate to function, this makes a lot of sense, right? Because it has a similar function, which is polymerization, adding nucleotides to a growing nucleotide chain, right? So why can it only synthesize telomere repeats? Why doesn't it copy the sequence of the DNA? Why is it only these repeats? Yes? Right, because it has this RNA template inside the enzyme. So that's what it's going to synthesize, only that sequence, or the complement of that sequence. Right, and that's why there are repeats, because it goes, and then it goes again, and it goes again, and so you have these repeats of these however many base pairs. Great. Would human telomerase, yes? Would these be considered non-coding regions? I would say they're very much non-coding regions. Yes, very much non-coding. Would human telomerase be able to elongate mouse telomeres? Well, the real question should be, it depends, right? So what do you guys, so do you guys want to talk to your friends a minute about this one, and then we'll come up, because there's a lot of quizzical looks on faces.
Pretty before that they have a different That might be a different species that they're coming.
Okay, guys, what do we think? What do we think? Do you think human telomerase could elongate mouse telomeres, and why? So I see nodding now. Why?
Usually, human stuff never works in animal stuff. Animal stuff works in human stuff.
Okay, so there's just that species don't line up. Okay, I want more science than that. Yes.
It depends on what the telomerase sequence might say. If it's PTA, PGG,
Depends on what the telomere sequence is in the mouse. Because remember, telomerase has its own template, but if it can bind, you know, if it has those same matching bases at the end, why not? As long as it can bind. Let's say it's different, then I probably won't bind. My guess, you know what, I don't know, we have to look up, but I have to say mouse and human are pretty close. Depends though on telomere repeats, I have no idea. Anyone want to add to that? Yes?
You have to change the surface proteins. Would it tag it certain ways so you're allowed to track it?
We're okay. So would you have to do something to it? So antibody? Are you talking autoimmune disease or something like that? Okay. So we're talking experimentally in a tube, not in a person. You take chromosomes out of, or you take some synthetic DNA that you make that has telomere repeats in the sequence of mouse, in the sequence of human, and you put it in human telomerase and see what happens. See if you can get extension. Yes, absolutely. If you put a mouse protein in a human, you're gonna cause an immune response. Absolutely. Yes. Could you manually change the template in telomerase? Gene therapy. I think. Why would you want to? Yeah, so it's kind of like when you talk about. So the question is, can you manually change the template in telomerase if there's some kind of mismatch and you wanted? Let's say you have cancer cells, and the cancer cells have mutated their telomere repeats at their ends so that telomerase no longer binds. Well, then it wouldn't be a cancer cell because actually telomerase activity is advantageous for cancer cells. That'd be a great treatment. Mutated the other way. But I think if you ever want to fix any gene, you're having to get rid of what's broken in the cell and then put in new, and that is fairly complicated. Because it goes into the category of gene therapy where it's all about delivery. And in the past, people have used retroviruses to do this and caused cancer in people. So I don't know how much progress has been made there. Okay, so what is a reverse transcriptase? Does anyone know? Yes.
It goes from RNA to DNA.
RNA to DNA. So would you say telomerase is a reverse transcriptase?
Yes.
Yes. Right? Because you have an RNA template and you're making DNA. Right? Have I told you anything about proofreading in telomerase? I believe there's no proofreading in telomerase. No. Remember, DNA polymerase has a actual domain that has exonuclease activity. It has this proofreading ability. So if the wrong base gets added, the strand shifts into the exonuclease domain, and the wrong base is removed. I believe this is not present in telomerase, which leads me to my next question. Are you likely to accumulate mutations in your telomeres? And what might be the outcome for the cell? Yeah. Yeah.
It wouldn't be detrimental because they're not included.
So think about it a little bit more. So the question I got was maybe, but it might not be detrimental because it's non-coding. Yes.
I think it might be because the DNA polymers probably won't be able to bind to it and finish that end of the
So you're saying that if there was a mutation, telomerase wouldn't be able to bind, or DNA polymerase? Which are you talking about?
when after the polymers elongates the leading strand, the DNA polymers will not be able to bind to it.
Think about that, and we'll reconvene. You wanted to correct your answer? Okay, so if there's a mutation and your telomere repeats, then probably your telomerase won't be able to bind because it's all dependent on matching up between the template that's in telomerase and your telomere repeats. So I think it could be detrimental. Yes?
I have a question. These are non-coding regions, and why is it such an issue that it can't cause cancer?
These are non-coding regions. Why is it an issue that what can cause cancer?
That this elongation of your telomeres can cause cancer.
It's not that it causes cancer, it's that it helps to maintain cancer. Because when you've got cancer, cancer is many things happening at the same time, right? Dysregulated S phase, cyclins something with cyclins, maybe something with signaling, RB, so many different things happen at once that basically force the cell to go into S phase in a dysregulated way. And we just talked about how highly regulated it is. And then also a lot of things happen usually that result in apoptosis not to be activated when it should be. The telomerase thing is that if you're going into S phase over and over and over again, if you don't have high telomerase activity, your chromosome is going to shorten. You're not going to be a very good cancer cell, right? If you have high telomerase activity, it's going to maintain those ends, and you're going to be a fantastic cancer cell. Yes.
So, how does DNA polymerase recognize the telomere?
How does DNA polymerase recognize the telomere region? So probably what happens is there's some proteins that are involved in recruiting DNA polymerase to the telomere ends to extend them. I don't know the exact mechanism, but there is something that's going to recognize this extended region here. A DNA polymerase and primase will be recruited, a primer will be made, and it will be extended. Yep, I'm not 100% sure exactly what recruits that next DNA polymerase. Any other questions? So we can make that an extra credit. What recruits DNA polymerase to the extended telomere? Because you're right, it has to be recruited somehow. Okay, last one. What happens to the lagging strand? Why did I say that? What happens to the lagging strand? Oh, that was just for me to make sure you guys understood that DNA polymerase comes in and extends it. Okay, so here's your T-loop. That's what it looks like. So remember, we have this overhang. So once this is extended, and then DNA polymerase comes, so let's go back again. Remember, DNA polymerase comes and fills that in. You still have this overhang here. And I've had a few questions. What happens to it be attacked by nucleases? And so what happens is it actually wraps around and invades the double helix and binds within the helix. And I'll show you. So this is what it looks like in real life. You can't really tell much from that. It just looks like a lasso. That is how it looks, okay? So here's your DNA telomeres, here's your 3', here's your overhang. So that was your shorter lagging strand, right? And so here's this huge overhang that what it does, here, let's see. Just follow it around. It actually invades inside the, see here, this should be your normal double helix, and see how this part is opened up, and it invades and goes inside the helix. You do not need to understand the exact mechanism. Yes?
Is it usually that?
It's huge apparently. It's apparently huge. I had no idea, right? I thought it was much smaller too.
Is that like a cell type that has a lot of telomerase activity? There's variation.
So is this a cell type that has a lot of, I don't know actually. Well, let's think about that for a minute. Is that a cell type that has high telomerase activity because there's variation? Well, really what you're dealing with here is the what's happening to this little overhang. It's pretty small here. Right, and that should be, that's the kind of dictated by the length of your primer, right? In my understanding, I think the whole thing does loop around and invade, because you can see there's this double-stranded region that's coming around. It's not just a single-stranded. So it's kind of a confusing structure, but just understand it kind of loops around and tucks in. And so that you don't have that single-strand region. Yes?
Is there a set place that comes up?
I have no idea. Is there a set place that does that? I've got no idea. There are a whole bunch of proteins that regulate this process. It's a whole process. And it's supposed to be related to the strand invasion that's involved in come on, guys. Yeah. Okay. I'll get back to you in a minute. But either way. Recombination. God, people. Remember recombination? You guys all learned about it in 309, and there's this strand invasion. Whatever. It's supposed to be similar process. So you'll notice here there's all these proteins on the telomere. So these are your telomere binding proteins. And you could think of your T-loop as kind of creating a cap upon which all these proteins bind, and the whole purpose is to create this stable end, and you're distinguishing the DNA from broken DNA, which activates the DNA damage response. DNA damage response results in cell cycle arrest and apoptosis, so you do not want to do that. So this is the way that cell kind of prevents that from happening. If you look at these proteins that are bound to it, it's really interesting for me. So these TRF, this is the major telomere binding protein. And then all of these, RAD, MR11, MBS, RAP, COO, those are actually all DNA repair proteins. And so I'm not sure what they're doing there, but it's interesting to me that they're all recruited to the telomeres. Yeah, so that's how your telomeres look. Any questions? Isn't this fun? Yeah? Okay. People are smiling, so that's good. No one's crying. Okay. So in addition to DNA, so now we're moving on. Say bye, telomere. See you later. In addition to DNA, protein components of the chromatin must be duplicated. So now is where you say, oh my God, I thought we were done with talking about this. What kinds of proteins are on chromatin? Histones, right? So we're coming back. All the proteins that are required for chromosome structure or function, they all need to be duplicated, right? You're not just going to have naked DNA sitting there after S phase. All the epigenetic modifications, methylation, it all is copied. Okay, epigenetic inheritance. You guys all knew this, right? Okay, so the way it works pretty much is surprisingly simple, but maybe it's just simplified in the textbook. Parental histone segregation. So you have the parental nucleosomes, and as the replication fork moves forward, they're just randomly redistributed between the daughter DNA molecules. So here's the replication fork, and as DNA polymerase and helicase and all those things move forward, these come off and then assemble. That's the big picture. Come off and they assemble on the daughter strand of DNA. Okay, to get a little bit more detailed. So what you have is the parental H3, H4 tetramers. Those randomly distribute to the daughter DNA. So some go to one, some go to the other. And the H2A, H2B dimers are removed. Remember, you have a histone octamer. So part of it randomly distributes to daughter DNA. The others kind of just hang out in the area. And then they're actively put back on. Some histone chaperones, mainly NAP1 and CAF1, bind to and load the H2A, H2B, and then also new H3H4 onto the daughter DNA. So all you're doing is disassemble, reassemble, right? So you can get bogged down with the details, or you can just realize, okay, they get taken off the parental DNA and added to the newly replicated DNA, and there are these proteins that are called histone chaperones. Whenever you hear the word chaperone, you have to think it's a protein that binds to something and helps it to do its job. Kind of like a chaperone at a school dance. Kind of. Not at all. So here, right, so here you have polymerase and your sliding clamp, and it's moving forward, and you get these coming off, and the green are the H3 and H4, and they're getting randomly distributed. And then also, remember, since they're randomly distributed, only half as many H3, H4s are going to be on either daughter DNA, and so you have to have synthesis of new H3, H4, which you should automatically be thinking, oh, so gene expression must increase for these genes around the time of S phase. And actually, if you look at, or maybe right before, if you look at when gene expression for certain genes are turned on, some, I think the histone genes tend to be some of the only ones active during S phase, which makes sense. So CAF1 is going to bind to those and load them on, and then NAP1 is going to be loading on the H2A, H2B dimers, and just reassembling them. So here's another way to look at it. The H3H4 tetramers are actively transferred by CAF1, and there's another cochaperone that's mentioned in another textbook called ASF1, and they're transferred from parental to daughter DNA. Histone tail modifications are maintained, and then new histones will assemble adjacent to the parental histones. So you have this random distribution of your histones, but then you get the other ones filled in. And then the parental histones recruit enzymes that will maintain the modifications on the new ones. Yes. Right? Because you need to have more histones being made. Well, so remember, okay, so you're saying transcription during S phase, what does it do when replication is coming to that region? Remember, origins fire, very different timing, right? So I don't know for sure, but my guess would be that histones either fire very early or very late, depending on, I don't know, we should look it up. But my guess is that segment of DNA that's encoding those histone subunits probably is replicated very late. I don't know, late or early, but one of those two, right? Yes?
So if all the histones are coming from the parent strand to the daughter strand, where does the parent strand get its histones from? Why, like, if it's...
From the dog?
Well, if it's taking all its histones out, does it just not have histones anymore?
Okay, ask that again.
So if it's giving all of its histones to the daughter strand, and there's none in the parent strand, does it just not have anyone after?
Well, where does the parent strand go during DNA replication? What does it become? Does it just disappear, or does it be... Remember, semi-conservative replication. Parent strand, daughter strand. So that parent strand is gone, right? Because it becomes the daughter strand, right? So don't think of it as, when I talk parent and daughter, what I mean is parent to daughter, like that. Okay? So you've got them down here, and then they're going to go like that. Make sense? So it's not like parent-daughter. That make sense? Yeah. Okay. I thought so. I was like, I better draw on board. Yes.
Okay, so it's those setup is not gonna be duplicated later on because now the parent here has two histones and here you only in the middle cell only have one histone.
Great. So what happens, right? So each daughter duplex of DNA, each daughter duplex has half as many histones, right? Because it goes from one parental duplex to two daughter duplexes. And that's what I'm talking about over here. What happens is you get, so here's random distribution, and then here you get newly synthesized H3 and H4 tetramers. And now I'm guessing you'll also get new synthesis. So you're not only taking and moving, but you have to get new synthesis. And if you think about it, as you go through cell cycle, you need more of everything, right? Each time, so it's like if in the beginning of the life of this cell type, there were 10 nucleosomes. You wouldn't last very long if all you were doing was redistributing, right? So you constantly need more.
So I just want to make straight. So every time we have DNA replication, there will be transcription going on.
Yeah, there's gotta be. Yes.
So when DNA is replicating and the chromosomes are being replicated, you said that the histones are divided and one goes to one point and another goes to the other point, and then later they're put back together.
Yep. So remember you got an octamer, right? And so your histones come off and they disassemble, and your H3, H4 are just distributed to the new daughter DNA, and then your H2A, H2Bs are then added. So you have that reassembly of your histone.
So every time you have a new histone added to the old ones?
Yeah. So you can almost think of like half of your histones on your new DNA are new and half are old. It's random, so. Okay, great question, Sis. This is good because it means people are thinking. Yeah. Huh? Well, it says it's random. Well, let's go for it because I think somewhere I have something about that. Because, right, so CAP1, which is one of these histone chaperones, so it binds to the H3, H4 tetramers, and it interacts with the sliding clamp. And so there is some level of regulation. So because that's all happening at the replication fork, so it's coming off and then going to the new DNA right behind it. Now in terms of even distribution, it says it's random. But I guess all you need are enough so that these reader-writer proteins can come and recognize modifications on the histones and spread them. Okay, any other questions about this? I guess we have about 1 minute left. Right, so CAP1 and ASF1, the chaperones, they're recognizing the new histones because the new histones are acetylated. They have lysine acetylation. And CAP1 binds to the new H3H4 tetramers and interacts with the sliding clamp to target the tetramers to the DNA behind the replication fork. And they assemble H3H4 tetramer onto the nucleosome, into the nucleosome. And then the new histones are deacetylated after they're deposited onto DNA. So I want you to just for 1 minute, this last minute, think about how acetylation is affecting loading of these nucleosomes onto the DNA, right? Does that make sense to you that you would have them acetylated, added to the DNA, and then deacetylated? based on what you understand about how nucleosomes interact with DNA, how histones interact with DNA, acetylation is it's going to make a weak interaction with DNA. And when you deacetylate, it's going to make a tighter interaction. So it's kind of logical that it would be acetylated as it's being loaded on to the DNA and then deacetylated. Okay, any questions? And then we'll just continue later. Okay?
Is Cap1 acetylated, or is it just like the
The
new thing
chaperone.
is already acetylated?
It's already acetylated by a acetyltransferase. It's recognized, so it probably has a